Translation, an important step in the synthesis of proteins and gene expression. Brought to you by the Geniuses. In transcription, an mRNA molecule is made from a template strand in the DNA. mRNA is read 5' to 3' and it is read in sets of 3. These 3 nucleotide bases are called codons. During translation, this mRNA molecule conveys important information from a gene to the ribosome. Translation occurs in ribosomes found in the cytosol of a cell. Before polypeptide synthesis, ribosomal subunits, mRNA, and the first tRNA assemble to form a complex. To understand how translation relates to previous steps of gene expression, it is necessary to understand the complementarity of codons in the mRNA and anticodons in tRNA. A tRNA molecule has an anticodon sequence in its second loop region that is complementary to the 3 nucleotide sequence of codons in mRNA. This allows it to bind to codons in mRNA in an antiparallel manner and according to the AUGC rule. tRNA molecules also carry an amino acid. An amino acid is bound to the 3' end of tRNA by an enzyme called aminoacyl tRNA synthetase. This enzyme catalyzes the attachment of amino acids to tRNA molecules. A tRNA molecule that carries an amino acid is referred to as charged. Cells produce 20 different aminoacyl tRNA synthetase enzymes, one for each of the 20 distinct amino acids. As a result, the 2 main functions of tRNA is to recognize a 3-base codon sequence in mRNA and to deliver an amino acid specific for that codon. This adapter characteristic of tRNA molecules is key to the synthesis of an amino acid chain from a mRNA molecule. My partner Jay will now talk about the 3 steps of translation in greater detail.
The 3 stages of translation are initiation, elongation, and termination. During initiation, initiator tRNA, mRNA, and both ribosomal subunits bind together to form a complex. The initiator tRNA binds to the start codon, and thus the amino chain begins. The stage that follows is elongation. After the complex is formed, more tRNA bind to the ribosome at its A site according to their anticodon, and link additional amino acids through the process of translocation using peptidyltransferase. A charged tRNA binds to the A site, and a peptide bond is formed between the amino chain bonded to the tRNA in the P site and the tRNA in the A site using the transferase. After the amino chain is transferred to the tRNA in the A site, the ribosome shifts one codon in the 3' direction and causes the translocation of the tRNA from the A site to the P site, and the now uncharged tRNA in the P site to the E site. This entire process repeats itself until the stop codon is read. Once the stop codon is reached on the mRNA strand, no more amino acids bind to the amino chain and it is released from the ribosome. This is the final stage, termination. The stop codon is recognized by a release factor that binds to the A site. This causes the release of the now unbound tRNA and amino chain from the ribosome into the cytosol. The final step of this stage is the disassembly of the mRNA strand, ribosome subunits, and the release factor. The components of the complex can be used again later for more translation.